Merci. Bonjour tout le monde. Merci beaucoup pour votre présence ici. C'est avec plaisir que je vous présente Dr. Lindsay McCulloch. Le nom de famille, dire pour moi, je ne sais pas, mais bon, j'essaie, ma prononciation, c'est. En tout cas, donc elle a fait son doctorat à Brown University et son postdoc, c'est tout postdoc à Harvard et aussi à l'Université de Floride. Floride, en fait. Donc, elle va nous parler de sa recherche, donc je pense que je suis content de l'avoir. J'espère que vous allez profiter de la conférence. Merci. Thank you.
Thank you. Thank you for the introduction, and thank you for the invitation to come here today to tell you all a little bit about some of the things that I've been thinking about. So my talk today is titled Exploring Missing Pieces of Our Understanding of Symbiotic Nitrogen Fixation from Ecosystems to Individual Plants. So I want to spend some time thinking about some weird paradoxical findings from various different scales about nitrogen fixation. But to take a step back, this is a picture of a tropical forest. It's lush, it's green, it's warm. We see lots of diversity of different plants here. And above ground, we can see a lot of these really charismatic mutualisms that we think about when we think about how a forest functions, right? We can see pollinators, we can see seed dispersers, but a lot of what has motivated my work is thinking about the mutualisms that are happening below ground. So thinking about how these mutualisms that we can't see easily with the eye might have these really big impacts on how ecosystems function now and into the future. So my work has really focused in on plant microbial mutualisms that are important for acquiring nutrients, so both bacterial and fungal. We have mycorrhizal fungi, where about 80% of all vascular plants form relationships with arbuscular mycorrhizal fungi, and then nitrogen-fixing bacteria, which are not as widespread in the plant kingdom, but some of our nitrogen-fixing plants are really important for our agricultural fields as well as for how our ecosystems function. So today I'm going to focus on just one of these, nitrogen-fixing bacteria, but I'm happy to chat about some of the work I've done with mycorrhizae as well. So the relationship that I think a lot about is symbiotic nitrogen fixation. I think about it in the context of legumes and their nitrogen-fixing partners. So symbiotic nitrogen fixation is the major way in which nitrogen enters most terrestrial ecosystems, or at least the most natural way. And so it's really important because nitrogen limits how much plants can grow. And so this mutualism can really regulate ecosystem level productivity. So here I'm going to show a lot of diagrams that kind of look like this, where we have our legume cartoon, and then these yellow circles are indicating these root nodules where this symbiosis takes place. And so just to give us a breakdown of how this works, we have our legumes. They provide carbon both in the form of these structures, these root nodules, as well as sugars to our nitrogen-fixing bacteria. And those nitrogen-fixing bacteria provide a source of nitrogen directly to the plant. And this is a huge advantage that these plants have because most of the nitrogen on Earth is found in this form, where you have two nitrogen atoms connected by this triple bond. So what does that mean? It means it requires a lot of energy to break that triple bond and make that nitrogen into a form that most organisms can use. So the way I like to think about it is all the organisms on Earth or in terrestrial ecosystems are bathing in this form of nitrogen, this really valuable resource, but they can't access it. So sometimes we like to describe this as being out at sea on a boat surrounded by salt water. You need water to drink, but you can't drink that form of water, right? So that's where these bacteria come into play because they've evolved the ability to form or to create this enzyme nitrogenase that's able to break this triple bond and form types of nitrogen that are more biologically available for most organisms. And this is provided directly to the plant. So if we go back to our metaphor, this is as if legumes have this ability to have a straw that they can turn salt water into fresh water, but just for themselves. So this creates a huge advantage for legumes in a lot of different ecosystems to have their own private source of nitrogen fertilizer. And most symbiotic nitrogen fixation in the tropics is thought to be facultative. And so that means in areas that we have high soil nitrogen availability, we expect that legumes are not going to invest a lot in this mutualism because there's already a lot of nitrogen in the ecosystem. Where if we grow the same plant in low soil nitrogen availability, what we expect to see and what we see in a lot of the agriculture literature is that we see an upregulation of symbiotic nitrogen fixation in these low soil nitrogen environments. So they invest more below ground with this bacteria mutualus in order to have that source of nitrogen when nitrogen is potentially really limiting. And the reason we think that this regulation occurs is because symbiotic nitrogen fixation is really carbon expensive. It takes a lot of carbon from the plant to do this. So to put numbers on that, about 16 grams of carbon are needed to get 1 gram of nitrogen through nitrogen fixation, where if a plant is investing in taking up nitrogen directly from the soil through its roots, it only needs about 4 to 6 grams depending on the form of nitrogen. So this is anywhere between 4 to 2 times more expensive in a carbon perspective for the plant. So we expect that plants are only going to really do this when it's really necessary. And so given that the carbon cost of symbiotic nitrogen fixation and that it can be regulated by the plant, we as a field kind of hypothesize that high nitrogen environments, 2 different things. One, that we would see a low abundance of nitrogen fixing plants in those ecosystems. They're going to be outcompeted by plants that don't have this carbon cost, or this is not as much of an advantage. So when we have low soil nitrogen availability, we would have high nitrogen fixer abundance. And when we are at high soil nitrogen availability, we expect that there would not be a lot of nitrogen fixers in that ecosystem. And we'd also expect kind of a similar pattern to happen at individual plant scale. So we would have low nitrogen fixation rates where soil nitrogen availability is really high. And when there isn't a lot of nitrogen in the environment, we expect that individual nitrogen fixation rates would be high. But that brings me to the 2 paradoxes that I'm going to talk about in my talk today. So first, nitrogen fixer abundance and diversity is very high in the tropics. And that's not what we would expect based off of those hypotheses I just laid out. And then we also see under high soil nitrogen conditions that often symbiotic nitrogen fixation is not completely downregulated. Sometimes it's a little lower, but it's still happening at rates much higher than we would expect. Given that it costs a lot of carbon, it doesn't make sense that plants are getting nitrogen this way. So if we start with our first paradox here, why is this paradoxical? Well, the tropics are not generally thought to be nitrogen limiting. So here in this global map, we see it's colored by what the ecosystem should be limited by when we're talking about soil nutrients. So the tropics tend to have this blue color. We think that the tropics are more phosphorus limited than nitrogen limited. And then kind of as you go up in latitude towards the poles, we think these ecosystems tend to be more nitrogen limited. And a lot of this has to do with the age of the soil. So nitrogen kind of accumulates over successional time. So when we think about ecosystems pre-disturbance, and that might even be thinking about glaciers, we think that nitrogen availability is really high in the ecosystem. You might have a disturbance occur that removes a lot of the nitrogen, and then it should accumulate over time until you get back to pre-disturbance levels. And this accumulation over successional time is a product of nitrogen fixation. So we tend to think that nitrogen fixation is really low in intact tropical forests. If we have a disturbance that occurs, we would see an upregulation of fixation at the landscape scale, and then it kind of is downregulated to these low levels once we're back at our mature forest. So again, given kind of this logical flow of ideas, we expect that tropical forest, since fixation is really low, that we would see something like this. Nitrogen fixer abundance near the equator is going to be low, and near the poles it's going to be high. And that could be a linear relationship or maybe something more like this. But we might expect that nitrogen fixing legume abundance is low in the tropics and increases where nitrogen availability is lower. And that's not what we see. We see that legumes make up 10 to 50% of neotropical forests. So these are our tropical forests in Central and South America. And we see the opposite pattern. So the number of legume species in these different forest plots is really high at low latitudes near our equator, and it just gets exponentially lower as we get closer to the poles. And we also see this paradoxical relationship that the amount of soil that's available in these same sites, soil nitrogen increases, and so does the number of legume species. So it kind of leads to this head scratching paradox of why are nitrogen fixers increasing in diversity and abundance towards the equator where nitrogen is thought to be abundant? Why are they not being outcompeted, or why is this not being selected out for when there's already a lot of nitrogen in the ecosystem? So if we go back to this figure, we have nitrogen availability is thought to be high. It's a product of nitrogen fixation, and that decreases as forest succession is occurring. And we think that what we're measuring when we go out to the field and we find our pristine, mature tropical forest, we measure 0 or very little symbiotic nitrogen fixation in our tropical forest, which is what we would think based off of everything I've already told you, right? But what we also are measuring is a lot of nitrogen leaching out of those ecosystems. So nitrogen is coming out through the streams, it's leaving our tropical forests, which if we assume that mature forests are at a dynamic steady state, or that the amount of nitrogen that comes in is about equal to how much comes out, we have a bit of a mismatch. And this is kind of an assumption that we take a lot in ecosystem ecology, that mature forests or ecosystems that have gone through succession are at dynamic steady state. There are things going in, and it's about equal of what's going out. So we're measuring a lot of nitrogen outputs. Where are the inputs? Maybe in reality, what we're missing is reality looks more like this, where nitrogen fixation is higher than we have been measuring. And so we have this potential mismatch between what we're measuring and what's actually happening in the forest. So what might explain this mismatch, right? Well, it's probably likely that nitrogen is not evenly distributed across the landscape. So here, this is a figure that's been modified by Menge and Levine, but it is getting at the idea that soil nitrogen availability is patchy in the forest, right? So this is a modeled 10 by 10 meter plot, and you might have some areas that have high nitrogen availability in our warmer colors, and then you have low nitrogen availability in our warmer colors and high nitrogen availability in our cooler colors. So this is a theoretical modeling framework. But what might cause this patchiness that they've modeled in a natural tropical forest? What I'm going to try to convince you is that canopy gaps in mature forests are probably what's causing some of this patchiness to occur, right? So when we imagine a mature tropical forest, we imagine these big trees, a very intact ecosystem, but disturbances are a normal part of how these forests function, right? So we have large trees falling all the time. They often bring down other trees with them. And so these canopy gaps in mature forests might be these unmeasured hotspots of symbiotic nitrogen fixation, so causing nitrogen fixation to happen at really high rates in this one area as the forest recovers. And we think that that might be happening because gaps have a higher nitrogen demand, so the plants that are there or around there have now the desire or the need to grow very quickly into this light gap. Light is very limiting in tropical forests, so they're going to need nitrogen to grow quickly and not get shaded out by others. And there's also a lot of light availability. So having more access to light, plants can photosynthesize more. They have that carbon they need to invest in this mutualism. So this disturbance dynamic that's happening might potentially favor upregulating symbiotic nitrogen fixation in these environments. And there are some compelling preliminary data that suggests that. So a couple of different studies from different parts of the neotropics find that when you look at both nodule number and mass where the symbiosis is happening, in mature forests it's really low, and in adjacent gaps it's elevated and much higher. This figure is showing different plant species and the percentage of the individuals that they checked that had nodules when they were growing in gaps, which are in orange, compared to forest. And it's not all the species, but the majority of the species, you find more nodules when they're growing in gaps. And this is really important because trying to understand how much nitrogen is being fixed in these ecosystems has implications to how we model carbon dynamics and make future predictions about our climate. And so this one paper, again, has primary nitrogen fixation or our mature forest basically at 0. And they had a similar thought that we do, that when you include gap dynamics, you're going to see elevated nitrogen fixation. But all of this is based off of modeling scenarios, right? So there's actually no measurements of nitrogen fixation rates in these gaps compared to the surrounding intact forest. But it could, they suggest, lead to almost 5 times more nitrogen fixation. So in order for canopy gaps to be hot spots for nitrogen fixation, we either need the likelihood of nodules existing to be greater in gaps compared to forests, and or we need the nitrogen fixation rates to be higher in gaps compared to forests. So I'm going to try to address this big picture question. Are canopy gaps in mature forests these unmeasured hotspots? And these are kind of the sub-questions to get at that. Are nodules more likely in gaps? Is symbiotic nitrogen fixation higher in gaps? And if so, what could be the landscape scale effect of canopy gaps on nitrogen inputs for these ecosystems? So to address this question, we did a large field observational study at La Selva Biological Station in Costa Rica. This is found in the northeast lowland region. It's a wet, hyperdiverse tropical forest, though it does have one nitrogen fixer that is very common. It's found here. It's also at the lower limit of one of the last full elevation gradients found in the tropics, which is seen here. So this is a really fun ecosystem to work in. What we did is we identified 25 canopy gaps on the landscape, and we went out and created these 8 transects to sample along. So each of these points is every 3 meters, where we sampled a whole bunch of things, both in the gaps in blue, this transition zone, and then the near the intact forest. We measured all different variables, potential environmental controls on nitrogen fixation, as well as trying to estimate symbiotic nitrogen fixation itself. We did this with the SACS method, which is you go along the transect, you take a root core or soil core, you look for root nodules. If you find them, you continue to take soil cores until you don't find them anymore. We do this because nitrogen fixation is thought to be clustered in the landscape. So to get a good estimate, you kind of do it this way. And what we did when we found nodules is we incubated them with 15 N2 gas to get an estimate of the rate. So I spent 2 years of my life trudging through the rainforest, taking all these soil samples, carrying them back to the lab. And today I'm just going to focus on talking about these data and what we found. So are nodules more likely in gaps? We actually find that we had a higher probability of finding nodules in the forest compared to the gaps in our first soil core. But again, when we look at our y-axis, we're talking about 4% versus 2%. Finding nodules is extremely rare in the landscape. It's a lot of zeros in your data. So there is elevated probability of finding nodules in the forest compared to gaps, but they're always kind of rare on the landscape. And on average, when we did find nodules, we found a lot more cores that had nodules in our forest compared to the gaps. So that kind of eliminates our first option, that nodules are more likely in gaps compared to forests. So if we look at our second option of symbiotic nitrogen fixation rates being higher in gaps compared to our forests, what do we find? Well, first, our nodule biomass is higher in gaps compared to forests. And then we were interested in looking at, okay, so we found nodules, and now we're going to see how much they're fixing. So this figure here is showing the amount of nitrogen fixed per gram of nodule, so how efficient those nodules might be. And we don't see a big difference between the forest and the GAP sites. So nodule biomass was on average about 6 times higher in the GAPs compared to the forest, but the nitrogen fix per gram of nodule doesn't vary between forests and GAPs. So it's not that nodules are magically more efficient when they're growing in a GAP compared to forests, there just is probably more of them. And so what we find is that nitrogen fixation, or the amount of the kilograms of nitrogen fixed per hectare per year, is higher in GAPs compared to forests. It's about 7 times higher, and it's mainly driven by differences in the amount of biomass of nodules there is than increased efficiency of those. So we find some evidence for this, that symbiotic nitrogen fixation rates are higher in GAPs compared to forests. And so what might be the effect of that on the landscape scale? So to just do some back of the envelope calculations, what we did is we took our landscape scale nitrogen fixation estimates without considering the GAP effect, so using just our forest samples, and we estimate that there's about a little less than 2.4 kilograms of nitrogen per hectare per year being fixed, and then using estimates of how much of the landscape is made up of GAPs versus not GAPs, we estimate how much our GAP effect might elevate our overall nitrogen fixation rates. And so this maximum GAP effect is based off of this paper from the 90s that suggests that 7.5% of the landscape is GAPs, which would lead to a little less than 20% increase in our estimates. And now there's more literature that's been coming out in the past year or so that suggests that probably tropical forests are made up much more of GAPs, especially as disturbances are increasing in our tropical forests. So this number is potentially higher. And some of my collaborators and I are working on using remotely sensed data to get a more accurate representation of the percentage of GAPs in this particular forest where we took these measurements. So measuring symbiotic nitrogen fixation in canopy GAPs may start to close this mismatch that I brought up earlier, where we have these measured things, but they don't really meet what we would expect based off of all the other information we have about this ecosystem. Because for a really long time, people avoided sampling in these parts of the ecosystem because they're very difficult to work in, and they're not what people imagine for an intact forest, right? So given that tropical plants face high competition with each other for light, for nutrients to survive in this tropical forest, and nitrogen availability is not evenly distributed, and having your own source of nitrogen might be a competitive advantage to taking advantage of these disturbance regimes, nitrogen fixers might have a competitive advantage here, and maybe that's why we see them maintaining this high abundance and diversity in these forests. So maybe one missing piece of this puzzle is really considering the spatial heterogeneity of nitrogen dynamics in tropical forests. I'm not the first person to think about it this way. There are other people thinking about it, but I guess my contribution to this subfield is thinking about it explicitly in canopy GAPs, but this idea that there's a lot of heterogeneity in the tropics that might be contributing to that is not my own. So I've spent the first part of the talk kind of talking about this first paradox, and I'm going to transition into talking about this now at the individual plant scale. So we were thinking about ecosystems, now we're going to think about individual plants. And this paradox that we're seeing even under high soil nitrogen conditions, fixation is not being completely downregulated. So we've talked about this dynamic between these plants and the bacteria, that when there's high soil nitrogen availability, that we wouldn't expect plants to invest a lot in this mutualism compared to in low soil nitrogen availability. But what we're seeing in the data is that sometimes we have results that look like this. So why are we observing symbiotic nitrogen fixation maintained even when high soil availability is present? And if it's not nitrogen availability that's controlling symbiotic nitrogen fixation, then what is it? And I've talked a lot about different environmental factors, but I haven't talked a lot about biotic factors. So thinking about the organisms themselves that are performing this mutualism, so thinking about the plants and the bacteria. There is lots of evidence that the legume identity, the plant species, alters how much fixation can happen. So some species are better at this than other. There's lots of different papers that have shown that. I'm going to show some data from some of my work, which you'll see why in a bit. But so one study that I did in Costa Rica, I had 3 different legume species. And what I found was that one species, Stiphidendrum microsacium, fixed about 0.01 grams of nitrogen per seedling per hour. That's kind of what I'm going to reference everything to. Zehia longifolia fixed about 2 times that. And then our third species, Pentaclethra macrolova, fixed about 12 times that. So we see really large differences in the capacity to fix nitrogen depending on the plant species. And we don't really understand why. And a lot of people in my field, a lot of ecosystem ecologists or biogeochemists, we think about nitrogen fixation in this way, where we have our plants, we're starting to believe that the plant species might matter. We don't think a lot about the nitrogen fixing bacteria, who they are. We treat them as like a black box that carbon goes into and nitrogen comes out of. Even though those are the organisms in this relationship that are forming this enzyme that's actually fixing the nitrogen. And lots of other fields have recognized that the bacteria identity is really important for how much nitrogen fixation is happening. Here with soybeans, the percent of nitrogen in soybeans that's coming from symbiotic nitrogen fixation depends on which bacteria strain you expose it to. And sometimes there's 2 times differences in the amount of nitrogen from 40% almost up to 80%. So the bacteria themselves probably matter a lot. Feel like I don't maybe need to convince this crowd of that, but some people do need to hear that. And so the questions for this part of this large project were trying to understand if there are unique amplicon sequence variants or ASVs or ESVs or unique sequences of bacteria or bacteria communities that might be associated with specific legume species. So are super fixing legume species great at fixing nitrogen because they have really good partners? Or is there something else happening? And then are there specific ASVs that are associated with higher or lower nitrogen fixation rates? So when you have this one bacteria present, does that mean you're going to be able to fix a lot more nitrogen than without it? Or is it kind of a community story? So to answer this question, I took nodules from the seedlings of those 3 species I told you about. These seedlings were planted bare root in the forest in these meter by meter subplots. So they were planted together in the same forest soil to be exposed to what they would normally be exposed to instead of in a greenhouse setting. And I used DNA metabarcoding to characterize the nodule bacteria community composition. And I did that looking at 2 different gene regions. So I used a broad spectrum marker of 16 sRNA and then a nitrogen fixation specific NIF-H marker. And so we decided to use these 2 different markers to kind of have a better understanding of what is in our root nodules. So when we collected the samples, we took them back from the field, we cleaned them, we surface sterilized them, and extracted the DNA. And so our broad spectrum marker might give us some really big picture understanding of the bacteria that are present. We might get data that look like this, where we have our relative abundance of different genera or order of bacteria. And then our nitrogen fixing specific marker might tell us less about the whole bacteria community, but these fine differences between bacteria that have the gene to fix nitrogen. So are the bacteria communities specific to our individual legume species? What we find when we do a permanova is that there is some separation happening amongst our legume species, but there's a lot of overlap. So the different colors are different host species. The points that are closer to each other have more similar bacteria communities than those that are further away. And so we see some clustering happening for our 16S broad spectrum marker, but there is a lot of overlap. and the same for our NIFH gene. And so, legume species identity explains about 14% regardless of the gene region of the variation in the nodule bacteria community. If we want to look at these data in a slightly different way, if we look at 16S, this is the relative abundance of the different bacteria orders of our 3 species. So we have Pentaclethra here, Zehia here, and Cypherdendrum here. And I think this is really interesting that we see there is a lot more diversity in the order of bacteria present in this plant that is fixing a lot more nitrogen than others. So also, when we look at the diversity, we find that Pentaclethra macrolova has significantly higher taxonomic and phylogenetic diversity in their nodules when we're looking at 16S compared to the other 2 species in our study. Where NIFH, we see a lot of mostly Bradyrhizobium. This is kind of what we expect to see in nodules, right? And this is what, based off of the literature, people expected that all of our figures would look like this, that there should just be rhizobia in our nodules and all these other orders shouldn't exist in there. And that's not what we find in a wild ecosystem. So rhizobia made up the majority of our NIFH ASVs in these legume species, and we don't see really big differences in the diversity, the various indices that we looked at. So are there specific ASVs that are associated with high or low efficiencies of nitrogen fixation? Because remember, we see this big differences in nitrogen fixation by plant species, but we also saw a lot of community overlap of the bacteria that were there. So are there individual ASVs that maybe are only associating with one species that gives it that outsized role or an advantage? And so we used a couple of different analyses. We use a species indicator analysis to try to see if there are particular ASVs that are indicative of really high or low fixation rates. And I don't have time to talk about all of those results, but the one that I find really interesting is we have this Actinospica, which was found in 83% of our Pentaclethra seedlings. It was about 10% of the relative abundance of reeds, and it was found in none of the other 2 species. And they're grown in the same meter by meter plot all over the forest, right? So those bacteria, this Actinospica is clearly all over the forest, but there is some selection happening because it was never found in our other 2 species. And when it is found in Pentaclethra, we have nodule efficiency here on the y-axis, and whether this Actinospica was present or not, we find higher nitrogen fixation rates. Now, I'm not suggesting that the Actinospica are fixing nitrogen, but something is happening that when they're present in the nodules, they're fixing, those nodules are fixing more nitrogen on average than when they are not. So again, they're not the only bacteria that are present, but when they are present, they tend to fix more nitrogen. So what's happening here? Not totally sure. I think it's kind of an interesting result, especially because for a long time in the literature, we just think about rhizobia and the efficiency of rhizobia. And my big, I guess, message I'm trying to get across is it might be much more complicated than that. And we might need to think more about the whole community. So maybe Actinospica is indirectly promoting fixation through plant growth, promoting hormones. And there's more and more kind of evidence of these kinds of things in the literature with wild legumes from Mediterranean climate. So I think we're kind of starting to unravel this piece of the puzzle. So nodule bacteria identity tells us some part of the story, but it still seems like we're missing something. It's not clear that just one or a couple of bacteria are really going to tell us whether a plant's going to fix a lot of nitrogen or not, or fixing more nitrogen when there's already a ton in the ecosystem. So are there other important environmental variables? What are we missing here, right? So one thing that we've talked a lot about is how much nitrogen is in the soil, but another thing that might influence how a plant allocates its resources is how much light that ecosystem has. So what we're starting to see more and more is that light is regulating symbiotic nitrogen fixation more than soil nitrogen availability. So these columns are the different light treatments of this one study. Again, these are a couple different studies that all show the same thing. And the x-axes of these individual plots are the amount of nitrogen fertilizer that they added. For context, 40 grams of nitrogen per meter squared per year is a lot. That's more than most agricultural fields will be adding. So we see that in low and medium light, nodule biomass, the percent of the plant biomass that is nodule is really low or zero. And the percent of the nitrogen that the plant is made of that's from symbiotic nitrogen fixation is also around zero. But then when these plants are grown in high light, regardless of the nitrogen fertilizer they're receiving, they're fixing a lot of nitrogen. And so some of my work has shown that. Others' work that is not only in the tropics, but also in some temperate regions have shown that. So there's this really compelling evidence that the amount of light a plant has might override whatever's happening with soil nitrogen availability. Well, what might explain that? There's this surplus carbon hypothesis that has been kind of growing in popularity. So the idea is that you have these various environmental factors that might lead to leaf growth declining, but photosynthesis continuing to happen. So sometimes when you have nitrogen limitation, or if temperatures are cold, moderate drought, other phosphorus limitations, the plant stops growing, but it continues to photosynthesize. And that means that there's a surplus of photosynthates or sugars that are accumulating in the leaf. They become these non-structural carbohydrates, and the leaf needs to get rid of them in order to continue to function properly. And so they are often shunted through the phloem, and these organic compounds are then distributed below ground in the roots, potentially for root growth, potentially as storage. This paper suggests that maybe it's going to mycorrhizae or root exudates. And so the plant is not investing below ground because it wants more below ground resources, but because it needs to get rid of this resource in order to continue to function. So it's kind of a shift in the framework of which we might think about this. And then that has all these implications for what's happening in the soil, with the soil biota, what the microbes are doing, which microbes are favored, and the soil organic material. And so what myself and some others have suggested in this paper we have in review is that, well, maybe some of these organic compounds are going to nitrogen fixation. Maybe we can apply the same framework to explain these paradoxical findings that we're finding with symbiotic nitrogen fixation. If that's explaining it mycorrhizae, maybe it's also works for nitrogen fixation as well. And maybe highlight should be added up here. And to just like give you kind of a visualization of how we theoretically are thinking about that, you might have a relative rate of whatever process you're interested in. Here we talked about it in this paper about drought. So the idea is that when plants start to experience low to moderate drought, they stop growing. So this yellow line starts to decrease, but they continue to photosynthesize for some period of time. And then this light blue shaded area is where we would have surplus carbon happening. And what we're suggesting is that surplus carbon is being moved out of the leaf material through the phloem below ground, and it's being invested in its below ground symbionts, not because the plant is trying to invest in it, but because it's trying to offload a resource that it needs to get rid of. And that might then allow the plant to have an advantage later on. So another potential explanation of this second paradox is that although nitrogen is the product of symbiotic nitrogen fixation, it might not be the ultimate regulator of this mechanism. So we might need to think a little bit more creatively and maybe collaborate with more plant physiologists to think about what might be pushing and pulling resources within a plant, and think about it a little bit less like this direct exchange for carbon for nitrogen, because maybe plants are not deciding things in that way. So to just wrap up, I talked to you all today about these two different paradoxes that have kind of been my head scratchers of my career so far, of why are these things happening? And so for the first one, nitrogen fixer abundance and diversity is high in the tropics. I think heterogeneity in the nitrogen demand and supply may give tropical nitrogen fixers an advantage, and that's why we see a really high diversity and abundance of these N fixers. And then I think we have to ask ourselves, are we looking in the right places? Are we assuming that nitrogen fixation is happening here and not there and not sampling there enough? There's also lots of other organisms that fix nitrogen that we might not be quantifying, like cycads, termites, all of these other things in the ecosystem that we might need to expand where we're looking. And then for the second one, under high soil nitrogen conditions, we don't see that it's downregulated the way that we would hypothesize based off of theory. So our bacteria partners are much more diverse than we think based off of the agricultural literature, and they're probably not passive players. They're probably not just in there taking carbon and pushing nitrogen out, right? So there might be multiple bacteria that are influencing the functioning of those nodules, and nitrogen fixers may just be responding to excess carbon. And it's not a response to nitrogen limitation or demand. It might just be a physiological response that is not linked to what we originally hypothesized. So I guess some key, very broad takeaways that I have is that we use these simple frameworks and principles to help us understand really complicated systems, right? And to ground our questions. But we should dig deeper when we keep getting these results that break our expectations. Sometimes we think, oh, well, that's just an outlier or something went wrong with that experiment. But if we keep seeing this happening and it's growing in the literature, maybe we should, think about what are we assuming? What are we assuming about our system that may not be true? So that's kind of, I think, the big picture things that I would like for you to take away from this talk. And I want to just thank the many people who helped do this work. This is like very, very intensive field work, and those plots don't look like a lot of data, but there are a lot of things that go into them. And all my funding sources, and yeah, the mountain of people who helped me do this work. And I think I have time to answer any questions. And my email's there in case you want to send me your thoughts. Thank you.